Okay, it's now 1:30, so I'm going to start on this webinar. We've got a few people in here. I'm David Mail from the Open University. I've been Professor of Biology for a while. I'm an immunologist and have a particular interest in a variety of conditions, but particularly also diseases of the brain. But this particular talk is going to be about viruses and vaccines. And in particular, I'm going to be concentrating on the recent COVID-19 pandemic and the things that we have learnt from it. So the things I'm going to go through are, first of all, let me put on a laser pointer as well before I start. We're going to go through how viruses infect cells, a little bit about how the immune system protects us against virus infection, a little bit on epidemiology and particularly what are called the reproduction number of an infection, the R values. We'll talk about herd immunity threshold and critical immunization level. These are related concepts about how many or what proportion of people in the population need to be immune to a particular infection in order for it not to spread during in the population. We'll then talk a little bit about the design of vaccines, how viruses can evade immune defenses, and finally, what we have learned from the COVID-19 pandemic. The COVID-19 pandemic was caused by a virus which is called SARS-CoV-2, which stands for Severe Acute Respiratory Syndrome Coronavirus number 2, and that is what produced the disease. So that's what we'll concentrate on. Very simple outline of how acute viruses infect cells. That we've got a cell here and a virus indicated by this sort of dark blue up at the top here. The first thing that a virus has to do to infect a cell is to attach to the surface of the cell, and it does this via specific molecules on the surface of the virus and on the surface of the cell. Then the virus has to infect the cell, which is enter it. It then starts a cycle of replication. This will vary between different viruses, but essentially the virus takes over to the cell's metabolic machinery in order to create more viruses, usually hundreds or even thousands of viruses from a single cell. Then virus is released from that individual cell so that it can potentially infect more cells. And finally, the overall effect of the reproduction of the virus and the release of the virus is that you have what's called cytopathic damage, which is damage to these cells or killing of the cells. So that's an outline of the basic process. I want to talk next about this first step in the process, which is attachment. And I'm going to talk about it in relation to the SARS-CoV-2 virus. This is an electron micrograph of the SARS-CoV-2 virus. It's a coronavirus. You can see why it's called a coronavirus because it has a sort of appearance on the outside of a little bit like a crown. These viruses are around about 50 nanometers across, so they are much, much smaller than a cell. And these spikes that you can see on the outside are the important spike protein which allows the virus to attach to the cell that it's going to infect. On the right-hand side of this slide, we've got a diagram of the internal structure of the virus. The center of the virus contains a single strand of messenger RNA, or RNA, which is the virus's genetic information. And that genetic information will be replicated as the virus replicates. Round the outside of that is an envelope which is derived from the plasma membrane of the cell that virus previously infected, but it does contain virus envelope proteins, small envelope protein and an envelope protein. And then, as I said on the outside, the very important spike protein or glycoprotein. A glycoprotein is a protein with carbohydrate units attached, which is on the outside of the virus. So how do viruses attach to cells in the first place? Well, as I mentioned, the SARS-CoV-2 virus has a spike protein on its outside, and it binds to a receptor molecule on the surface of various cell types in the body called ACE2, which is angiotensin converting enzyme 2. And that molecule is present on lung epithelium, intestinal epithelium, and renal epithelia. And that determines which cells the virus is going to be able to infect. If you look at influenza, it has a protein on the outside called the hemagglutinin, which allows it to attach to sialic acid residues, which are on the surface of many cells of the body. If we go down to the bottom, rabies virus, which, as you know, infects nerves particularly, has a surface molecule, glycoprotein G it's called, which binds to the acetylcholine receptor, a molecule that is present on nerve cells and muscle cells. So the attachment of a virus determines what we call its tropism, or which cells in the body it's going to be able to infect. Now this diagram gives an outline of the key antiviral defenses that protect us against infection with an acute virus. By an acute virus, I mean one that comes and goes, like flu or colds or SARS-CoV or COVID-19. Here's a virus. It's going to infect this particular cell here by attaching and entering and replicating. But as it does so, this cell will release a signaling molecule called interferon, which signals to neighboring cells in this tissue, and it induces virus resistance. It effectively shuts down protein synthesis in neighboring cells, and that means that should those cells subsequently become infected with virus, the virus can't replicate in them. This is a very important process in the early stages of infection because it limits the rate of spread of a virus. Now, antibodies is something that you will have heard about from the pandemic. An antibody is represented here by this Y-shaped yellow molecule. An antibody binding to the outside of a virus here can prevent it from attaching to a cell and therefore prevent the cell from being infected. So antibody is very important for preventing infection. But antibody also promotes the destruction of virus by cells of the immune system. But that is really only half of the equation, because we also have a group of lymphocytes in the body, white blood cells, which are called natural killer cells, and another group which are called cytotoxic T cells. And these cells between them are able to recognize virus-infected cells, and they then give them an instruction to commit suicide. So the cell will die if it's virally infected, but it was probably going to die anyway. And by killing the cell, it prevents the virus replicating and spreading. So that's an outline of the different types of antiviral defenses. Now, next question is, when do these particular antiviral defenses kick in? Well, for an acute virus infection, we're looking over a time course here of 14 days going on to a month and for a year. An acute virus infection, such as COVID-19, takes place over around about a week, 7 or 8 days. And during that time, the virus increases in number and then it's gradually controlled. In the first stage of an infection, interferon and those natural killer cells are particularly important. And they are just holding the line until the lymphocytes, the key cells of the immune system, T cells and B cells, cytotoxic T cells and B cells, which go on to make antibody, are able to sort of get themselves into gear. They are also replicating very quickly at this stage in order to put enough lymphocytes of the correct type into battle against the virus. So these cells will develop after about 3 or 4 days. But the important thing about them is that they persist for a very long time in the body, and they produce what are called memory cells, which effectively memorize that infection and will produce a much stronger and more effective immune response if that infection is encountered again. Now, these cells, the B cells in particular, start to produce antibody, which appears after about 5 to 7 days. An antibody is a molecule floating around in the blood and in the tissue fluids, which, as we've seen, can block virus infection. Antibody persists for a while, but eventually, after a number of months or years, it gradually declines, and so eventually it will become undetectable. But of course, we do have these long persistent memory cells which are able to reactivate and can subsequently produce the antibody again if needed. So that is when antiviral defenses actually take place. I'm going to move on now to talk a little bit about epidemiology of infection. And the first idea I want to introduce is something called the basic reproduction number, or R-nought. And this is how many people, on average, an infected person will go on to infect themselves. So we've got a population here of people which are indicated by this sort of light yellowy colour. Each one of these is an individual person. If we introduce a single infected individual, the purple person here, into this particular population, over on the right-hand side, that person has infected 4 other people. In this case, we say that R-nought, the basic reproduction number for this virus in this particular population, is equal to 4. And that's the basic reproduction number. Here are some R-nought values for various virus diseases. So influenza, which is a pretty unpleasant disease, has a reproduction number of between 1 and 2. It's not terribly infectious. At the other end of the scale, a disease like measles has a reproduction number, basic reproduction number, of between 12 and 18. So a single individual can infect a very large number of other people in a susceptible population. And there are numbers for a variety of infections here, but you'll notice that SARS-CoV-2, which is the one that we're talking about here, has a reproduction number between 2.5 and 6. And you might think that that's quite a big variation, but if you will remember that there were actually quite a lot of variants of SARS-CoV-2, and the different variants have different reproduction numbers, as we'll see later. Now, when we talked about the R-nought, we made a number of assumptions. The first assumption was that everybody in the population was fully susceptible to the disease. Now, right at the beginning of the COVID-19 epidemic, nobody had ever encountered the virus before, and it is true that everybody was fully susceptible. The other assumption is that the population is equally mixing. We call this homogeneous mixing. Now, neither of these things are generally true in the real world. So, for example, for most infections, some individuals will be partly or fully resistant. So for something like flu, people will have caught flu before, and some people will be resistant to it, and some people will be susceptible. So not everyone is normally susceptible in a population, unless you've got something like COVID-19, which is a brand new infection. The second point, which is also not true, is that mixing varies from one person to another. I mean, for example, if you had a bus driver will be meeting lots of different people as they come through the bus. And so therefore they will be meeting a large number of people. Conversely, if somebody is working at home and rarely going out and maybe only staying with their family, they will be meeting far fewer people and therefore mixing much less. So people in large institutions will be mixing with large numbers, people who are living by themselves with far fewer people. So in the real world, we talk about what's called the effective reproduction number, or RE, and this is often just called the R value. And when you heard those talks from Downing Street by Chris Whitty and so on, they would be talking about the R value, and this is what they were talking about. So RE is the reproduction number in the real world where some individuals are immune and different people are acting differently from other people. Now, the key point about this is that if RE is greater than 1, each person will be spreading the disease to more than one person, and in that case, the infection will spread and become an epidemic. Conversely, if RE is less than 1, the infection will gradually die out. And we can see this sort of illustrated diagrammatically here. Here's our population of people again. In this case, though, half of the population, which are indicated by these dark green spots, are resistant to the virus infection. We put our infected individual into this population here, the purple person, and that person would normally potentially be able to infect 4 other individuals. However, 2 of those individuals are resistant to the infection, and that means the actual transmission is to only these 2 susceptibles here. In this case, we have a disease where the R-nought value is 4, 1, 2, 3, 4, but the actual transmission, the R value, the effective reproduction number, is 2. Generally speaking, the effective reproduction number is going to be less than the R-nought value. And so to control an infection, what you need to do is to reduce the RE value. Now, how can you reduce the RE value? Well, if there is an increase in the number of resistant people in the population, that will do it. And that could happen by natural infection, or alternatively, it could happen because the people have been vaccinated. If you take a disease like measles, most people will not get a natural infection, and therefore the resistance in the population is provided almost entirely by vaccination. Now, if you look at the second assumption, how can you reduce the amount of mixing of the population to decrease the number of contacts between individuals? Well, that can happen either because of voluntary behavioral changes, for example, people distancing from each other or avoiding crowds. And this certainly started to happen at the beginning of the COVID-19 pandemic as people were very alarmed by it and were therefore keeping away from other people. But during the COVID-19 pandemic, in the initial stages, around about 40% of the people were asymptomatic. That is, they didn't even know that they'd got a disease or perhaps thought they'd got a mild cold, and continued mixing in the community as normal. And that meant that it was necessary to introduce compulsory changes, which was lockdowns and closure of public events and so on, in order to reduce the amount of mixing. Now, as time went on, and we're talking about a year or so during the pandemic here, there was a gradual move from controlling the RE value by reducing the number of contacts between individuals to controlling it by reducing the number of susceptible individuals in the population by vaccination. But at the beginning of the pandemic, of course, we didn't have vaccines, and so these reducing contacts was really the only option. Here are the RE values for COVID in the United Kingdom between June 2020 and October 2022. They're shown, each of these bars, as a bit of a range because obviously it varied between different parts of the country. But here's the critical R value of 1. Remember that if it's less than 1, then the infection is dying out, and if it's more than 1, the infection is spreading. You can see that in the early parts of 2020, the lockdowns that we had there caused the infection to reduce, R value less than 1. And then the brakes were taken off around about the autumn, and the R value started to increase. There was a further lockdown in the autumn. It started to decrease again. This is a very interesting little point here. Just before Christmas, the government decided that in order to get the shops moving and allow people out to get presents and that sort of thing, that they should allow people to go back into shops and start mixing again. And you can see there was immediately a big increase in the R value at that particular point. And just before Christmas, they locked down again in order to be able to control the epidemic. And you can see the way this varies around 1 as the various measures were put in place over the course of the year. Now, the R value is not the same as the incidence of the disease. This graph here shows the incidence of COVID-19 infection in the United Kingdom between April 2020 and April 2022. And you can see that at the beginning, actually, the numbers that were infected, the incidence, which is the number of new infections every week in the entire population, actually, sorry, this should be every day, is really quite low. We had a big outbreak in early 2022. By early 2022, a lot of people were vaccinated. The disease was less severe. But if you take this particular point here, you have a high incidence, but it's dropping. So the R value here is less than 1. Conversely, if you go back to this point here, the actual incidence is fairly low, but it's increasing, so the R value is high. So incidence can be high when R value is low and vice versa. They're showing different things. Next idea I want to introduce to you is what's called the herd immunity threshold, or HIT. And the idea here is what proportion of the population needs to be immune in order for a disease, an infectious disease, to die out. Here's our example again. This time we've got 12 individuals and 9 of them, dark green, are immune to the infection. They are not susceptible, they're resistant. And then we've got 3 people here, 1, 2, 3, in the lighter color, who are susceptible. So we could say that 3/4 of the individuals are resistant, that's 0.75, and 1/4 of the individuals, 0.25, are susceptible. Now, when our infected individual here enters that population, potentially they can spread the disease to 4 individuals. But in practice, only 1 of those individuals is actually susceptible. At that point, the R value, the RE value, is equal to 1, and that is the critical immunization. That is the herd immunity threshold. It's also, in some cases, the critical immunization threshold, as we will see. Now, I'm going to take a tiny break here, and for you to have a go at doing something. For a minute, I'm going to give you the chance to calculate the herd immunity threshold, or HIT, for a number of virus diseases. And the value is calculated by this equation here. 1 minus 1 divided by R0 multiplied by 100%. I've done this for SARS-CoV-2, the original Wuhan strain, which came from China, for which the R-nought value is 2.5. So 1 minus 1 divided by 2.5 is 0.4. So that is 0.6 times 100% is 60%. And then you can have a go at doing this for rubella, mumps, and measles, and see what herd immunity threshold is for those particular infections. So I'm going to give you a minute or so to have a go at that, and then I'll come back in with the answer, and we'll carry on talking about how you vaccinate against these infections. I'm also going to have a look at the chat box in case any of you have put any questions in there. Yes, Triveni, you are correct. Rubella is 80%. So I'm just picking up a message out of the chat box by one of the people who is listening in on it and has just done the calculation. Mumps 87.5%, also correct. You'll see that measles is the most infectious. Yes, it is. We've got it in, 93.75%. I'm just going to move over here, and there are the actual answers which you have come up with absolutely perfectly. I'm going to close the meeting, the chat box now, and carry on. So you can see there that effectively the larger the value of R-nought is, the bigger the herd immunity threshold. At the beginning of the pandemic, because there was not much option, people said, oh, well, let's wait until we get herd immunity. It would have been necessary for 60% of the population to have caught coronavirus at that stage in order to get to herd immunity. Not only would that have been very serious and we would have lost a lot of people due to severe illness, but the hospitals would have been completely overwhelmed at that stage, as they were in Italy in the early stages of infection. So next idea is the critical immunization threshold. So what proportion of the population must be vaccinated for a disease to die out? For a disease such as measles these days, only a small number of the people catch the disease and acquire natural immunity. Immunity is mostly acquired by vaccination. And in that case, and for COVID-19, before most people had caught it, the herd immunity is the same as the critical immunization threshold, because that's the only way you get any immunity, by vaccination. Here are the critical immunization thresholds for a variety of virus diseases. So flu with the lowest R number, you need 50%. Measles with the highest, you need 94%. And this is the measles, mumps, and rubella, as you know, come as a set of childhood vaccines. And if you don't get that high proportion of vaccination in the community, then first of all you will see measles spreading, then you will see mumps spreading, and then you will see rubella spreading as the level of immunity in the overall population gradually drops. And so that's why it's important to keep vaccination levels high. Now we're going to go on to talk a little bit about how you deal with an infection by vaccination. And the basics of this rely on an essential component of the immune response, which is that a second encounter with a foreign material, which is an antigen, produces a stronger immune response than the first or primary encounter with that antigen. Okay, so this is a time over days. At this point here, day 0, a person gets a first challenge with an antigen or a first infection, and then you get the appearance of antibodies. There are 2 different types of antibodies here, immunoglobulin M and immunoglobulin G, and they sort of start to arrive after around about 7 days. Now, after the second encounter with the antigen, the first thing that you see is that the lag time between the production of the challenge and the production of the antibodies is much reduced. So the lag time is shorter. The second thing is that there's a much higher production of antibodies, particularly this immunoglobulin G, which is more effective than IgM, and it is more effective for a number of reasons, but one of them is that the binding, the affinity of that antibody for the thing that it's binding to is much higher, and therefore that makes it more effective. Also, a key thing about this second part of immune response is it extends over time for much longer. It doesn't die off like the primary immune response there. Okay, so this is the basics of why we vaccinate, because the secondary immune response is much more effective than the first response. So we vaccinate here so that if you encounter the real infection here, you have a flying start on your immune system. Here's the basics of vaccine design. Vaccines are made in a number of ways. Here's our virus, which contains the virus's genetic information there. There's the virus's sort of capsid, and on the outside are the proteins which allow the virus to attach to the cell that it's going to infect. Ways of producing vaccines: you can produce a live attenuated vaccine. In other words, a virus which is alive and can replicate, but it doesn't produce pathological damage. And for example, some of the polio vaccines are in this type. Another way you can produce virus is by using an inactivated virus. You effectively kill the virus so that it can't replicate. And some of the polio vaccines are like this, and some of the earlier rabies vaccines were like that too. Third way you can produce vaccine is you can take just a component of the virus, say for example the protein on the surface which allows it to attach to cells. That's the spike protein in the case of SARS-CoV-2, and introduce those into an individual so that they make an immune response, produce antibodies against the viral components without actually encountering the whole virus. Now, that was the traditional way of making vaccines up until the time of the COVID pandemic. And then what they did was they started to use genetic ways of producing vaccines. You take the virus's genetic material, the virus genes, and you can introduce those genes into a harmless virus, what is called a viral vector, and that produces a vector vaccine. The vector vaccine is of the type that was produced by AstraZeneca early in the pandemic. Another way of producing this is to just take the RNA which encodes the viral spike protein and use that RNA, getting it inside the cells of the human body so that the body itself makes the viral proteins which are then recognized by the immune system. And these are what are called DNA or RNA vaccines. They use just the genes. Here are some examples of COVID-19 vaccines taken to clinical trials. More than 100 vaccines were developed in laboratories, and only a proportion of them, maybe 20 or 30 of them, were taken through to clinical trials during the COVID pandemic. But all of them were really mostly concerned with producing an antibody response against the spike protein, which is illustrated on the left here. It sits on the membrane of the virus, and that is the part of it there that actually binds onto the cell that it's going to infect. So the Pfizer vaccine, the Moderna vaccine, and one by a company called CureVac used the messenger RNA, which encodes this spike protein. The Oxford AstraZeneca vaccine was a vector vaccine containing the gene for the spike protein, as was the Russian Sputnik 5 vaccine. And then a couple of other companies, Johnson & Johnson made one in this vector vaccine type, and the Chinese produced one from CanSino Bio, their company there. Interestingly, these new ways of producing vaccines produce vaccines more quickly than the old traditional ways of just using virus components like the spike protein, which GlaxoSmithKline did, or using inactivated virus, which various companies like Valneva and a Chinese company, Sinovac, did. So there were a variety of ways of producing vaccines. It turned out that the newest ones were the most effective and also the fastest to get going. Producing an RNA vaccine, though, is not just a question of taking the genetic material of the virus and putting it into the human body. You have to get it inside the cells of the human body so that it will actually generate the spike protein. And this gives you an outline of the complexity of producing these proteins. Here is the RNA here. First of all, you have to shield it and protect it because RNA is really quite a delicate molecule. And so it is protected with a lipid with polyethylene glycol on the outside. And then you have to get it inside the body. And this is done by mixing it with lipids in ethanol and gradually raising the pH. And what happens as the pH is raised, these lipids, mixture of them, form micelles, which trap the virus nucleic acid in the center. So it's now protected, and it also has a coat on the outside, which is similar to the phospholipid bilayer of the human cells. And this then is readily taken up by the cells. So you've got to not only protect the RNA, but you've also got to get it inside the cells. The RNA vaccines, though, are still relatively delicate and required very low temperature cryopreservation. Otherwise, they would effectively break down. Whereas something like the AstraZeneca vector vaccines are altogether more robust and did not require such low temperature storage. So there are advantages and disadvantages to each of the different types of vaccine. How do you measure antibodies against SARS-CoV-2? You can do this by using a technique called ELISA, or enzyme-linked immunosorbent assay. And the plate at the bottom shows this assay as applied to the serum samples that have come from the blood of a single individual who has been vaccinated. Here's what's going on at the top here. For this particular individual, on day 0, they had a serum sample taken from their blood, and then they were vaccinated. Subsequent samples were taken on day 7, day 14, and day 21. And then just before they got a vaccine boost on day 56, another sample was taken, and then another sample 2 weeks after that, and finally a sample at day 200 in order to see how long the antibodies lasted. Okay, you're probably not familiar with an ELISA plate, but if I show you this is day 0, day 7, day 14, day 21, day 56, day 70. And these are the dilutions of these sera. Essentially, when you see red, it means that it can detect antibodies that are present at a significant quantity. So at day 0, you can see that there are no antibodies. At day 7, you're starting to get the appearance of antibodies, and you can detect them at a serum dilution of 1 in 4. By day 14 and day 21, the level is up to 1 in 16 or 1 in 32 dilution of serum. But immediately after this boost at day 56 and going up to day 70, you can see a big increase in the amount of antibody, going up to a dilution of 1 in 256. And importantly, these antibodies persist for quite a long time, right up to day 200. So once you've started testing out your vaccines on people, how effective are they when you start looking in larger populations? And this graph is what's called a violin plot. And in a violin plot, this shows you the range of antibody concentrations that are detected as against, in this case, SARS-CoV-2 spike protein. And the dot in the middle is the median value, and the width of it indicates the proportion of people in the test sample who have got that particular level. So if, for example, you take the AstraZeneca vaccine in people aged 65 to 84, the range of titers is between 1 in 10 and 1 in 10,000. The median here is around about 1,000, and most people have antibodies in this sort of 1 in 1,000 kind of range. If you compare that with the Pfizer vaccine in people aged 65 to 84, you can see that the range is between 100 and 100,000. The median here is around about 10,000. And this shows you that the Pfizer vaccine is producing more antibody than the AstraZeneca vaccine in this particular trial. Interestingly, down here on the right-hand side, we've got a population of people who were not vaccinated, but did catch the infection naturally. And you can see that all the people who were vaccinated have higher antibody levels than people who got their antibodies via natural infection. On average, there is a range here. I'm not going to go through this calculation with you or ask you how to do it, but if you are looking at this on a recording, you might like to have a go at this, how to calculate vaccine effectiveness. In this particular example, we've got 17,762 people who are vaccinated and 8,550 who are in the control or placebo group. In the vaccinated group, 73 people became infected. In the placebo group, 130 became infected. And you work out the proportion that became infected in each group, which is that calculation there. And then you work out the vaccine effectiveness according to this equation here. And if you do that, you will find that the AstraZeneca vaccine, this particular one, was 73% effective, which allows you to compare effectiveness in different trials. Different trials are not usually done exactly the same way, and they're in different populations, so they're not fully directly comparable, but it does give you an idea of how effective a vaccine is. Now there we measured vaccine effectiveness according to whether a person got infected. But of course, we were actually much more concerned with a number of other things, like whether a person became seriously ill, whether they became hospitalized, whether they were actually died as a result of the infection. And the one thing that vaccination did was to move this overall profile of disease over to the left. Remember that a significant proportion of people, 30 to 40%, were asymptomatic even in the very first stages of the COVID-19 infection. Most people had mild illness, some had serious illness. It was these people here that were hospitalized and the danger of overwhelming the health services that was really the concern. And the vaccination shifted the whole of this sort of profile over towards the left. Here's how the SARS-CoV-2 vaccine coverage went in the United Kingdom between January 2021 and October 2022, indicating the proportion of people up to 100% here who had a first 1 dose, 2 doses or 3 doses of the vaccine. And you can see that we needed about 60 to 80% immunity in order to stop that particular Wuhan strain of the virus spreading, and that by around about April to July of 2021, we did actually achieve 60% of virus coverage in the UK against the initial Wuhan strain of the virus. However, what happens when a virus mutates? Well, there is the danger that it evades the antibody. So by the change in the shape of the spike protein, it means that the original antibodies can't fit on so well to block infection. Now, there were a variety of variants of concern arose during the pandemic, and they were given designations alpha, beta, gamma, delta, and so on, Omicron, by the World Health Organization. And here are the R-nought values for some of these variants. So the original Wuhan strain has got an R-nought value of about 2. By the time we got round to the Omicron strain, turning up in 2022, the R-nought value had gone up to 8.2. The viruses are getting more infectious. They have to get more infectious because the resistance level in the population is gradually increasing, and therefore the R-nought value has got to increase in order for the virus to be effective as far as the virus is concerned. And you can see that some of the variants that we had less concern about, epsilon, for example, have relatively low R-nought values. Now, just because a virus is more infectious doesn't mean that it's more pathological, doesn't mean it produces worse disease. And indeed, many of these later variants, although they were more infectious, potentially able to spread quite well, they were actually producing less disease, partly because people were resistant and partly because the virus in some ways was adapting to the human population. So here's a question. Do new variants evade antibodies against older variants? And in this case, we have the serum samples from 3 individuals, COV47, COV72, and COV New York. So these are 3 different antibodies. And this is looking at the infectivity against the dilution of plasma, their plasma, which is able to block infectivity. So for example, down here with this particular person, a 1 in 1,000 dilution of their plasma is able to block infection, whereas a 1 in 100,000 dilution of their plasma is not able to block infection in a laboratory. Now, in this case, they're looking at infections with either the original Wuhan strain, which are the black ones, WT, or with some of the variant strains which came later with particular mutations in their spike proteins here. Now, for these 2 individuals, you can see that their serum is more or less equally effective against the original Wuhan strain and the subsequent strains. But for this person here, his serum is pretty effective against the Wuhan strain, but not nearly as effective. He needs a much higher concentration of serum to block infection when you're looking at some of these other variant strains. So the answer to this question is do new variants evade antibodies? Well, it depends on the individual and what antibodies they have produced. Usually there is some residual immunity from antibodies, and there's almost always some residual immunity and protection from those memory lymphocytes, which make immune responses faster subsequently, particularly the T cells and the natural killer cells. I mentioned variants of concern. What do I mean by a variant of concern? Well, the World Health Organization defines these as viruses that spread more quickly, viruses that produce more severe disease, obviously, and viruses where the clinical symptoms are significantly different from the symptoms produced by previous strains of the virus. So how can you respond, or how can one respond to the appearance of new variants of concern? Well, here is a later variant of the vaccine, and this one is what's called a bivalent vaccine. It was produced by Moderna against COVID-19. It contains the original Wuhan strain, and it contains some of the Omicron BA.1 strain, the variant that arrived in 2022. The RNA vaccines can be adapted relatively quickly and reformulated by sequencing new variants of the virus and matching the vaccine to the gene sequence of the new variants. But one doesn't do this as sort of just a knee-jerk response to the appearance of new variants, because there are literally thousands of new variants arriving all the time. It's only worth producing vaccines when there are variants of concern which are more pathogenic or significantly different from previous variants. And the reason for that, of course, is it costs a lot of time and money and effort to get your vaccine factories producing the new vaccine, and it's only worth doing that if there's a variant of concern. So, by way of summary, some things we have learned from COVID-19. It's difficult to track and control an epidemic when a large proportion of infected people are asymptomatic. They tend to carry on mixing in the community. The original SARS, which arose in 2002, wasn't like that. It produced symptoms in everybody. It was easy to spot when a person was ill and confine them and treat them, and therefore prevent the spread of the infection. That wasn't the case with SARS-CoV-2, the COVID-19 pandemic. Public health measures can blunt the force of an epidemic to protect the health services from becoming overwhelmed, and they also allowed time for vaccine development. That turned out to be very important. A lot of people thought that the public health measures were designed to stop people ever getting COVID-19. This just doesn't happen. They were going to get it eventually at some stage, but by slowing the force of the epidemic, the health services could be protected and vaccine development did take place. We know that virus mutations often increase the infectivity of the infection, but not usually the pathogenicity. Residual immunity from previous strains helped protect us against later strains, so that the disease was generally less severe later in the infection or the pandemic when there are new strains arising. And the key thing from an immunologist's point of view is that the messenger RNA vaccine development was rapidly accelerated through 2020. It's widely applied to treatments for other conditions, such as cancer. And so we have genuinely moved forward in vaccine development. Now, finally, if you find that what we've gone through is interesting and you want to delve into it in greater detail, I have produced what is called an Open Learn course for the Open University. It's free. It takes around about 12 hours of study. It has a virtual laboratory embedded in it where you can do that ELISA assay and you can start examining serum samples. It goes through the virus mutation and development, the immunology in much greater detail, and do feel free to visit Open Learn at the Open University and find this course if this particular area is of interest to you. Thank you very much. I hope you've enjoyed the talk. If you would like to add some questions, we've got a few minutes more before the end. I will open the chat box on my own computer so that I can see what's going on. Please write into the chat box if you would like, and I will attempt to answer any questions that you might have. If there are no questions, I will say have a good afternoon.